name's Charis Vela. I'm a senior at UTD and I'm studying molecular biology. And we are in the Winkler lab. I'm studying under Dr. Winkler and Dr. Stephanie Boyd. And this project specifically is an aconitase-like enzyme critical for propionate catabolism as a new target for antimicrobial development against phytopathogens. Basically, our project, citrus greening disease, causes citrus crops to die all over the world, and it gives it this like reddish tint, and it gets bitter, and it doesn't really survive very well, and it affects all kinds of citrus plants. And the bacteria, Candidus libibacteria, is what causes this disease. It's a phytopathogen, so that means it like causes diseases in plants, basically. So our key target is an enzyme called ACND, which is used in the 2-methyl citrate pathway. And if we block that enzyme from working, then we have a buildup of propionate, which is toxic to bacteria. Basically, we stop the enzyme from functioning, we can kill the bacteria without harming animals, and we can use that as like an antimicrobial agent and stop citrus plants from dying. We're going to grow some overnight cultures, so to set them up we're going to use bacteria and glycerol stalks. We're going to use some M9 minimal media that only has exactly what we need for the bacteria, and then some glucose for our carbon source for the bacteria. And then we're going to turn on a flame for sterilization. We do 5 milliliters of the media. And now we're going to add the glucose. So since this is glass, you can flame it before you put anything in it to help sterilize it. And then we're going to do 100 microliters of the glucose. And then this one's wild type bacteria, so it's just normal bacteria in glycerol stock. And then we're going to scrape off some bacteria with a pipette tip and just leave it in there. So this is our shaker, so we can get air into the bacteria because the bacteria need air to grow, and it'll also incubate it at 37 degrees Celsius, so about body temperature. And then we leave them overnight.